Thanks,
Hi,
Michael.
thank you. Thanks, Charlie. It's great to see so many familiar names, and I think I got this responsibility honor to introduce Katia Koelle because I maybe know her longer than have known her longer than anybody else attending, just because we went to graduate school together at University of Michigan. I saw Randy here somewhere too. He was there when we were there, though of course he was a professor. But yeah, so Katia went to Stanford, finished in '97, and then got her PhD in 2005 from the University of Michigan, where she worked with the wonderful Mercedes Pasquale and on cholera dynamics. I think she was already, when I met her, when she arrived at University of Michigan, she was already, had already done quite a bit of infectious disease modeling. And as an undergraduate, and then in between her undergraduate and starting grad school, it's already pretty well established. She went on to do a postdoc with Brian Grenfell at Penn State University, and then was hired at Duke and received tenure at Duke. And then I think, if I remember correctly, she took a sabbatical at Emory University, and I guess really liked the taste of Atlanta city water or something. And so she moved to Emory University, where she is now as an associate professor. And that's about all I have on Katia's brief bio. I will say that she is sharing the floor today with one of her doctoral students, Michael Martin, and she'll introduce him when he speaks. Okay, Katia, take it away.
Thanks, Michael. Yeah, I just want to introduce Mike Martin first before we start presenting slides. Mike Martin's a fourth-year PhD student in my group in the population biology, ecology, and evolutionary biology program he started, of course, kind of before SARS-CoV-2 hit, and so his first, I guess, year and a half at Emory really focused on influenza virus genomic diversity, both within hosts and kind of between hosts, and then all that was kind of put on hold, and he's kind of one of these students who kind of just dropped everything and focused entirely on SARS-CoV-2, and even keeping up with the literature, which has been amazing because there's so much right now. So he's contributed quite a bit to the literature so far. So today we're going to be talking a little bit about some of our work reanalyzing a previous kind of finding in the literature on transmission bottleneck sizes. He's also contributed and really drove kind of perspective piece in science on how viral sequences, how SARS-CoV-2 viral sequences can be used and how they can be useful for understanding disease transmission patterns. And then he has also worked quite a bit with collaborators of ours at Tel Aviv University, and then also at University of Wisconsin, doing more phylogenetic analysis. And so trying to actually estimate epidemiological parameters such as R0 using viral sequence data. So he's been great to have in the lab in the last few years. And so he's going to be sharing the floor with me today. Okay, and it's good to see so many of you here. I was actually just finishing up my my evolutionary biology class syllabus for the class I'm starting to teach tomorrow, and for the discussion sections, there's a lot of you whose papers are being whose papers are on the syllabus. So yeah, it's great to see you guys here. All right. So I think everyone can everyone see this? Yes?
Yep, looks good.
Okay, great. Thanks, Meredith. All right, so we just have kind of a few slides prepared, few being about 15 or so, and some of them are kind of interactive. So that would be great if you can kind of interact, especially the graduate students among you. All right, so what we want to talk about today are transmission bottlenecks, viral transmission bottlenecks, and specifically SARS-CoV-2 ones, and why we actually even care about quantifying how large these transmission bottleneck sizes are. All right, so first of all, what are transmission bottlenecks? So if we have, okay, here we go. I'm gonna, hold on. Laser pointer, there you go. Okay, so what are transmission bottlenecks? So if we imagine during the process of disease transmission, right, we have a donor individual, right, and a recipient individual, and the virus is being spread from the donor to the recipient. And so you can imagine that the donor who's donating the infection has a relatively high viral load, right? And they might be coughing or sneezing, or maybe just even talking closely to the recipient. And so some viral particles are being transferred, right? And so what the transmission bottleneck size is, are the number of viral particles that actually kind of seed the infection in the recipient individual, right? So it's the ones that, the number that actually kind of establishes genetic lineages within the recipient host. And so we also talk about this in the context of NB, or the transmission bottleneck size. So it's not the number of virions that just kind of fall on, the recipient, and it's not the number of unique viral genotypes that establish infection, but it's the number of viral particles that establish genetic lineages in the recipient host. And so you can imagine that transmission bottleneck sizes can be very, very small, right? So only a few viral particles can get across and establish infection, or that they might be very large, right? And so we really have been interested in trying to kind of, quantify kind of how many, what that transmission bottleneck size is. Okay, so how do we actually estimate them? So there's a variety of kind of methods available in the literature. A few years ago, we've developed we did a review of the methods available, and we felt like there was a little bit left to be desired. And so we established our own kind of inference method by extending some previous methods. And so the way we estimate transmission bottleneck sizes is using this, what we call a beta binomial method that we developed and is published here in this journal virology paper. And it's been used previously in applications to influenza virus and more recently SARS-CoV-2 by us and a few other groups. So the basic idea here is that we have, again, this donor individual. That donor individual has a lot of virus in them. You can deep sequence that viral population. And then what you can do is using these short reads, you can call variants along the genome. And so you can see here, for example, these two sites shown here are polymorphic. And we have here this, we have at this site, we have two alleles, one which is kind of slightly above the detection limit here, the detection limit being kind of this dashed line. And then at this other site, we have kind of a higher frequency minor variant shown here in blue. So then you can imagine that in the recipient host, we can kind of do the same thing. We can sample that recipient host, deep sequence that sample, and call variants. Right. During the transmission process, of course, kind of there's a smaller amount of virus that's transferred, right? So this is the founding population size, or the transmission bottleneck size, and that this virus population actually grows until the time of sampling. Right. So how we estimate the transmission bottleneck size is actually using the polymorphic sites. So looking at the variant frequencies at these polymorphic sites in the donor and seeing what those frequencies are in the recipient. And if you're interested kind of more in the methods, I'll just point you over to this paper, or we can discuss it later. Okay, so why do we care about the transmission bottleneck size? So the transmission bottleneck size has implications for evolutionary dynamics and disease dynamics at various scales. And so I'll go into these in the next couple of slides. First, the transmission bottleneck size might have implications for within host dynamics, right? So what's the starting population size? And also kind of potentially the propensity for disease development or the severity of infection. It also has implications for being able to identify who infected whom, right? In these kind of transmission clusters. And then it also has implications for how quickly population level, the virus can adapt at the population level. Okay, so let's consider kind of this first one here within host dynamics and disease development. So there's been kind of discussion in the literature and from a lot of experimental studies that have looked at different inoculum doses, right? There's been oftentimes this tendency in these experimental systems, these animal models, that if you give the animal, a higher dose right, that they might develop more severe disease. So this is the case also kind of with SARS-CoV-2. This is some work out of Yoshi Kawaku's group, looking at these Syrian hamsters who are either given a low dose of the virus or a high dose of the virus. And here you can see kind of in days post-infection, just the weight loss, right? And you can see that the hamsters that were administered a higher dose of SARS-CoV-2 lost more weight, and also here had more severe symptoms, right? And so one question is are individuals who are getting more severe infections of SARS-CoV-2, is it potentially because their inoculum dose or the number of viral particles that started their infection might be higher than for other individuals, right? So that's been a question that's been asked in the literature. And so this relates to something known as the variolation hypothesis. This has been primarily put forward by Monica Gandhi and others. Variolation here is the idea that kind of started with smallpox. The idea that you can take a small amount of virus, from someone who's been has been recovering from smallpox, take a tiny little bit of amount of virus from like a scab, pulverize it, and then by either kind of shooting it up a nose or by kind of injecting into a small site, that individual can gain immunity but have very low chance of developing very severe disease. Right, and smallpox had a mortality rate of about 1 in 3, and so this variolation process was used to provide immunity with a much lower risk of death, right? And so you can imagine this is a smaller dose, right? And so there's been this idea put forward that maybe wearing masks, maybe, it's been demonstrated over and over that masks protect an individual from becoming infected, and masks also prevent others from being infected from someone who's wearing the mask, right? But maybe there's another possibility. Maybe masks can actually also impact the probability of developing severe disease, even if a breakthrough infection through the mask actually occurs, right? Because there might be fewer viral particles establishing an infection. So that's the variolation hypothesis for SARS-CoV-2. And so to evaluate that hypothesis, it would be useful to have an idea of kind of how many viral particles, even without masks, how many viral particles actually get across to see an infection. Okay, so transmission bottlenecks, of course, are also quantifying how large they are also important for seeing whether we can figure out who infected whom in a transmission cluster. And so you can imagine that if transmission bottleneck sizes are very large, right, and you have a lot of minor variants in an individual, that would help facilitate identifying who transmitted to whom within a cluster. But if transmission bottleneck sizes are very small, that's kind of compromised the ability to figure out who infected whom, because it's really the dominant genotype that gets transmitted in those cases. Okay, and lastly, transmission bottleneck sizes are important to quantify because if those bottleneck sizes are very loose, so a lot of viruses get across, then you can imagine that a beneficial mutation that arises in a donor is oftentimes then transmitted if bottleneck sizes are large, right? And so that variant could then also increase in frequency in the recipient, and population level adaptation could occur pretty rapidly. However, when transmission bottleneck sizes are tight, a beneficial mutation that occurs in a donor could oftentimes be lost during the transmission process, right? And so that kind of slows down population level viral adaptation. Okay, so given kind of these reasons for why we want to know what, the magnitude of transmission bottleneck sizes, what we were interested in doing is revisiting some work that was published in Science Translational Medicine. This was published in, I think it was early 2020, maybe it was January 2020, maybe March 2020. Later than that, it was published December 2020 or January 2021, but they actually looked at samples from February to April 2020 from Austria. And so what they concluded in this analysis was that transmission bottleneck sizes were on the order of about 1,000 viral particles. And so when we read this work, we found this interesting, in particular, in light of some really nice work that was done by Adam Loring's group at Michigan, which concluded that for seasonal flu, transmission bottleneck sizes were more on the order of about 1 or 2. So we were really surprised to kind of see that these transmission bottleneck sizes would be on the order of about 1,000. We reanalyzed these data, and before I pass it over to Mike to go into some of the results, I want to kind of just do a little bit of a thought experiment. So what you'll see are a lot of plots. We're here on the x-axis. We have the frequency of a certain allele in the donor, right? And then here on the y-axis, the frequency of that variant or that allele in the recipient, right? And so if there's a large bottleneck, let's say we have 2 variants, single nucleotide variants in the donor, one at this frequency and one at this frequency. If the bottleneck size is very large, what frequencies would we expect them to be at in the recipient? Anyone?
Similar or the same?
Yeah, great. I don't know who said that, but kudos to whoever said it. Yeah, exactly. So if the transmission bottleneck sizes are large, we would expect the recipient viral population to look a lot like the donor viral population, right? And so what we expect are kind of a scatter of points, right, along the x equals y axis, right? So that would be indicative of a large bottleneck. What about if the bottleneck size is very small? So let's say the bottleneck size is 1. What do we expect?
A lot of variability.
Okay, great. Anyone want to add to that?
Yeah, what you would expect is one allele to be lost and the other one to be fixed.
Yeah, exactly. So if the transmission bottleneck size is just 1, right, 1 genotype can get across, right? And so if there's a variant here in the donor at this frequency, right, that variant either has to be lost, right? So it should be over here or should be fixed, right? And the same thing over here for this variant, just depending on kind of which genetic background these variants, are in. And so if you have something between.
Are you assuming no selection?
Yes. So the assumption here is that it's just neutral genetic variation. Yeah.
Is that?
Yeah. There are some methods that have been kind of developed that have kind of tried to add on kind of address that assumption of all this genetic variation actually just being neutral, and they allow for the incorporation of
The other question is, are you assuming that donor and recipient have the exact same selection pressures on the virus?
Well, here we're assuming that there's no selection, so yes. But in these other methods that actually allow for selection, the assumption is oftentimes that there's the same selection coefficient between hosts. Yeah. Okay, and so if we have a small bottleneck size of 1, we would expect there to be just these variants in the donor to either be lost or fixed, right? And if you have something like 2 or 3 or 4 or 5, transmission bottleneck sizes of those, you expect kind of that there can actually be a genetic variation that's kind of transmitted, but it would actually fall kind of far from the 1-to-1 line in general. Okay, so I'm going to hand it over to Mike to go through some of first their results and then our reanalysis of it. If that's good for you, Mike.
Yeah, that's great. Can you, do you want me to share my screen? Or do you just want to you can just keep sharing yours.
Yeah, just let me know when I should
okay.
Switch it over.
Yeah, so these are their bottleneck estimates that they presented, and in the top is the figure that was in sort of the main text, and on the x-axis you have the different transmission pairs, and the y-axis is the estimated bottleneck size, the confidence intervals, and they're sort of colored by their family. So you can see that they estimated a really wide range of bottleneck size using a variant cutoff of 1%. But sort of what really piqued our interest here is that in the supplement, they presented this other figure using a slightly higher variant calling threshold. And you can see that the estimated bottleneck sizes change, but they change sort of not in an inconsistent manner. So the estimated bottleneck sizes overall becomes very bimodal. So you have a handful of pairs, these first two all the way to the left, where the estimated bottleneck size drops really considerably when you increase that variant calling threshold. And then some others, the green one that's about a third of the way across the plot, the light green, family 8, just a little bit to the left, Katia. Yeah, right there. That transmission pair, the estimated bottleneck went from just slightly above 10 to about 1,000, so there's clearly something going on where these variants between 1 and 3% are telling you something different than the variants above 3%. So we dug into them a little bit more. I guess, Katia, is there anything else you wanted to cover from this slide? Okay. So this is the plot of the variants from a single transmission pair on the left. So the x-axis is the donor, the y-axis is the recipient, and you can see that there's this variant here that's present in about 20% of the viral population in the donor that's completely absent from the recipient viral population. But at the same time, there's this variant that's present at, let's say 2% in the donor that was transmitted into the recipient. So that's sort of, highly unlikely that when you're sampling that viral population in the donor that you would not grab that variant that's in 20% of the reads, but just happen to grab a virus that has sort of this much less likely variant in it. And then, so this sort of indicated that these low frequency variants are telling a different story than the high frequency ones. And then on the right is sort of, we re-estimated their bottleneck sizes. This is essentially the same plot as we showed on the last slide, except the x-axis here, the transmission pairs are organized based on the maximum allele frequency in the donor. So that's the highest, the most probable, or the most common, or sorry, the sort of a degree of the amount of oh God, I'm stumbling on this. The variant that's the most common in the donor. And essentially what you can see is at the 1% cutoff, there does seem to be a correlation where when you have a high frequency allele in the donor, you're estimating smaller bottleneck sizes. So these are the black dots here, and you really estimate these very large bottleneck estimates are largely from the transmission pairs where you lack high frequency genetic variation in the donor. And then when we increase the cutoff to 3 or 6%, you can see that you get much lower estimated bottleneck sizes. All right, and then next slide, Katia, I guess. Okay, so we wanted to think about what these two stories were telling us. So I guess my question for you guys is, what do these variants that are sort of in low frequency in the donor, but shared between the donor and the recipient host, what story does kind of that tell us about the bottleneck size? So these variants that are sort of closely on this 1-to-1 line here. Come on. I don't have as much teaching experience as Katia does in getting people to answer questions.
Are the low frequency variants pointing to a larger bottleneck size?
Yeah, exactly. So the fact that they're shared between the donor and the recipient, even though they're at pretty low frequencies, and then even more so, the fact that they're shared at sort of almost identical frequencies, are both pointing towards a very large bottleneck. But then sort of on the flip side, that single variant at about 10% here, I mean, this is a different transmission pair, that is telling you sort of the opposite story, because that's not shared at all. And then finally, sort of what are all right. what is this point? So there's this variant that is sort of a new variant in the recipient. It's not identified at all in the donor. Sort of what does that tell you about sort of the bottleneck size and the early dynamics after transmission?
Well, there was a new mutation in the recipient right away.
Exactly. And not only was there a new mutation, but it rose to be fixed in the viral population. So the smaller the bottleneck size, the smaller the population size is going to be early on in infection, and then the more likely you'd get that fixed variant. Whereas if there's a large bottleneck, your population size starts off much larger, and it's much less likely that this variant would fix by drift over such a short amount of time. So you'd expect a new mutation to be sort of somewhere between 0 and 1, but less likely to be in all of the viral particles.
Can I just add one thing to that explanation, Mike?
Yeah.
Is, if this variant over here, that's a de novo mutation in the recipient and it got fixed in the recipient, and so it was introduced very early on in the viral replication dynamics and then fixed. It also means that if it only occurred once in a certain genetic background, right, and recombination rates are relatively low, right? It basically means that you can't actually have any of this shared genetic variation over here, right? Because this mutation must have occurred in a single genetic background, right? And so must have kind of, swept through the population kind of as a hard sweep, which again kind of points towards these very low frequency variants and their correspondence kind of in the donor and the recipient samples as potentially being spurious.
All right, so I think we can go to the next slide, Katia. So we were sort of wondering what these variants were that were shared between donor, recipient, host, and then shared at exactly the same frequency or nearly exactly the same frequency. So we sort of looked at these, and what we found is that there were a lot of mutations that were found across almost all of the samples. So here on the x-axis, each sort of strip here is a single mutation, and then on the y-axis is the frequency of that mutation in a given sample. And then you can see in the x-axis labels, they're a little small here, but we've labeled that with how many of the samples that mutation is found in. And there's only 43 samples total. So these two mutations are found in, these two low frequency mutations are found in every single sample in the data set. And then, and this is just I think the 10 most common, but there's very many of these that are shared between, large numbers of samples at, frequencies between 1 and, even above 5%. So this was sort of, I don't know, the piece of evidence that I find the most compelling, that these are almost certainly not genuine mutations. And even if they are genuine mutations, they're not variants that are being transmitted between the donor and the recipient host. If they're real mutations, then they are almost certainly being regenerated within hosts, because otherwise you wouldn't expect them to be in so many of the samples. And so this, when you're estimating the bottleneck, you only want to include variants that are shared, that are actually being transmitted, not homoplasies if you have them, or sequencing error, which these could be. So based on this, we kind of decided that we needed to increase the variant calling threshold, which is sort of a bit of a brute force method to filter these out, but we weren't really confident that we could do anything else that would leave us confident in low frequency mutations based off this plot.
Hey, Michael?
Yeah.
I'm sorry, I forget this paper. What sequencing technology did they use? Was this Amplicon, or was this the the enrichment panel, or what?
It was one of the early Arctic protocols. I think V2, but don't quote me on that.
Okay.
Yeah. So I don't know enough about, I'm not a lab sequencing guy, so I don't know enough about if these could arise from sort of PCR or from barcode swapping, all the above.
I mean, there are like these are known artifacts of, some of these are known artifacts of Arctic, so you guys nailed it.
All right. Yeah. Yeah, I guess that's good to hear. So yeah. So once we raised, and I know on the left we've already showed this, but you can see once you raise the variant threshold cutoff high enough, such that you get rid of those, you really are sort of across the board estimating really low bottleneck sizes. And unfortunately, you sort of lose out the ability to estimate the bottleneck for some of the pairs, because if there aren't any variants identified, you can't estimate because you have no data, and you get slightly larger confidence intervals because you have less data as well. But for the ones that we do have data, all the variants above 6% are pointing towards a really small bottleneck. And I think sort of to this point, this also like, I think in terms of like the bioinformatics side of things, it's like easy to think that, oh, if you just sequence something to a really high depth, you can call it really low frequency variants. And I think the more of this data that I look at, the more I realize that that's not the case. And there's like I said, I'm not a lab sequencing guy, so this is kind of out of my area of expertise, but I think there's a lot more that go into calling low frequency variants than just having high sequencing depth, because these data were really deeply sequenced, and you're still seeing sort of what seem to be artifacts. So because the confidence intervals were pretty big, we wanted to combine the data across pairs. So we sort of assumed a zero truncated Poisson distribution for the number of virions transmitted, and then sort of estimated the probability combining the data. And sort of based on this, we can conclude that I think 99% of transmission pairs, you expect there to be transmit between 1 and 3 virions. I think, Katia, is the yeah. So that is sort of our like single point estimate combining the data. So someone asked in the chat what the depth of sequencing was, and I don't remember exactly, but it was like, I think on the order of like 10,000 reads. So it was pretty deep. 10,000 reads like at average across the genome. All right.
So I know we're supposed to go into the discussion a little bit also. So maybe we should just kind of end there with which is kind of the end of our comment here. And so we think the transmission bottleneck size is extremely low for SARS-CoV-2 and similar in size to that of seasonal influenza viruses. And I know that Katrina Lithgow and Matthew Hall have a science paper from about half a year ago or so, where they also use the beta binomial method on some of their data, and they also conclude a very small transmission bottleneck size. And and there's a group that has more recently looked at Delta and has similarly concluded a very small transmission bottleneck size. So maybe for discussion, maybe we can open it back up to these three things that I mentioned at the very beginning about kind of the implications of transmission bottleneck sizes, right? And since we think that they're relatively low, and actually pretty tiny at this point, what does that have us, if we revisit kind of, the plausibility of the variation hypothesis, for example, or seeing whether we can identify who transmitted to whom in a transmission cluster, what are the implications for a small transmission bottleneck for that? Or what does that mean about kind of how quickly the virus can actually adapt at the population level? So maybe we can kind of focus on that, or we can focus on anything else you guys want to focus on. And I think, Vaughn, you have a question?
I do, but I feel like, so first I just want to say thank you. Great job, Michael, and a terrific intro, Katia, as that I sort of expect. You've left really kind of the juicy conclusions for us. So since I'm an insider, I'm not gonna kind of say much more, but I just want to say broadly as a complement of this approach is that it shows very clearly how what you'd think would be better methods can give confusing data that is both somehow consistent with a large bottleneck and a small bottleneck, and until you sort of think it through and take a hard look at the data itself. And so at once it's it's I think valuing a population genetic approach, but also you know, kind of a careful bioinformatic approach too. And of course, it's, you know, one of the challenges of our time. So bravo, it's going to be, this is the beginning of an awesome lecture for you, Michael.
Thank you.
Great, thank you. So it looks like we have a couple questions in the chat. A question from Kaylin Ryan and another question from Dana Hawley that seem related. So Kaylin was asking, does a low bottleneck size tell us anything about transmission more generally? What about masks? And then Dana was asking, do you expect that there's any relationship between exposure dose and transmission bottleneck size? For example, even if exposure doses are quite variable, is the transmission bottleneck likely always small?
Yeah, so I think some of that gets at this variolation hypothesis. You know, I think, and so, I mean, I think it's clear from these animal studies that inoculum dose has a big effect, right? But when they say low dose, they mean a lot of virus, and when they say high dose, they mean a lot of virus, right? That's being injected into the nose or wherever else. Right. And so if we're thinking about kind of the viral particles on the order of 1 to 3, you know, or maybe without a mask it would be 10 viral particles. With a mask, you know, maybe if there's an infection that results, maybe it's 1 or 2 viral particles. But I think in that range where you're, you know, where it's, you know, very small in general, right, that you have all these kind of, you know, very stochastic effects, right, early on, and that variation probably doesn't affect, you know, disease severity. So if I get infected from having a conversation with someone up close, you know, and I'm talking to them for 5 minutes versus 30 minutes, you know, or if they sneeze on me versus just kind of, you know, they have no symptoms, you know, my hunch would be that that the amount of virus that gets transmitted, whether it's, you know, 2 viral particles or maybe 10 viral particles, if it's very close contact, that doesn't really affect disease outcomes much, and that it's more a factor of, you know, age of the individual, host genetic factors, who knows what else, right? I mean, of course, kind of viral genetics in terms of, you know, which variant it is, you know, but that it probably doesn't have as much to do with, you know, how long you had that conversation, you know, or whether you were wearing a mask during that conversation. Again, I want to say, you know, if you're wearing a mask during that conversation, you're less likely to get infected, probably, you know, but it's, but whether it affects your disease, you know, outcome or your severity of infection, my hunch would be no, that there wouldn't be good correspondence there. I don't know if you want to add anything, Mike.
No, I think that, yeah, it was a good summary.
Excellent, thank you. So we've got lots of great questions in the chat. We do love to hear you physically ask your questions. So if you're willing to do that, feel free to raise your hand. But for now, I'll move on to the next question in the chat, which is from Terrence Taylor, who asks, how do your findings compare with similar work on other viruses, or more specifically on other coronaviruses?
Do you want to tackle this one, Mike?
Oh, sure, I suppose. I mean, I think Katia mentioned that this is very consistent with sort of findings from work that she's done with seasonal flu, that the bottleneck tends to be very small. I am not aware of any work on bottlenecks of seasonal coronaviruses. I don't know if it's out there. And then I do know for sort of other viruses, it can really depend on sort of the transmission pathway. So like for HIV, for example, I think there's work like where you can quantify the bottleneck comparing, you know, the different routes of transmission, and they can be quite variable and dependent. But again, I'm not actually, I don't know this off the top of my head.
Yeah, I think that's a really good point. There was a really nice study by Varble and co-authors out of Ben Tenover's lab at Mount Sinai, and they barcoded influenza virus, and then they looked at kind of transmission between different sets of small mammals, like, ferrets, or so they considered other small mammals as well. And then they looked at contact transmission versus, aerosol transmission. And depending on your route of transmission, there were a different number of barcodes that were transmitted. In any case, it was relatively small. It was on the order of about 10 barcodes were transmitted, or fewer. So that's consistent with Adam Loring's estimate of 1 to 2, bottleneck size. It's interesting for seasonal flu because there was actually a paper in, I believe it was Nature Genetics, that concluded also very large bottleneck size for seasonal influenza. But it turns out that that's some really great detective work on the part of Jesse Bloom and Catherine Shaw at Hutch, reanalyzed those results, and potentially because of chimeric, either some kind of technical contamination, like downstream code, and processing of the reads, or potentially formation of chimeras. It's probably I would guess the former. There was a kind of contamination of those data, and that if you actually look at Adam Loring's data, it's actually kind of more on the order of 1 or 2.
If I could just ask a quick follow-up, sexual transmission was brought up, these, contact, respiratory. What about vector transmission? Anything known about the bottleneck sizes with vector-borne viruses?
Yeah, there's some work also, and this is not exactly kind of my area, but I believe there's also been studies looking at barcoded virus potentially in Zika, and kind of looking even at kind of how bottleneck sizes actually, and what they are kind of within kind of a mosquito from kind of taking of kind of a blood sample, right, all the way through kind of the gut and so forth to onward transmission. And so it seems like there would be kind of bottleneck sizes there. And in general, there's, viruses that are transmitted in a vector-borne fashion, seem to actually, their substitution rates seem to be about an order of magnitude lower than directly transmitted diseases. And so that could either be, again, because of bottlenecks, through kind of that transmission cycle, or it could be because of, vectors and mammalian hosts, could have different selection pressures, so that's another reason why. But the little bit of work I think, I know of in kind of that area, also is seeing small transmission bottlenecks.
Great, thank you. Looks like we have a raised hand from Bernie Crespi. Bernie, you can go ahead.
Hi, yes, thanks for a fascinating presentation. It seems very odd to me that you can have presumably large variation in the initial dose of virions that someone gets, but then end up usually having only one genetic lineage that actually gets established. Is there any sort of process that we know about or could hypothesize that would result in a very quick selection within the initial establishing population of viruses? For example, one virion kind of gets the jump on the other ones and gets ahead of them, as it were, or any possible types of suppression. I just don't understand how you can have such big variation, presumably in number of, say, inhaled particles, but then only one usually lineage gets established. It doesn't seem intuitive to me. Seems like something's missing.
Yeah, that's really interesting. So one question is about kind of selection, right? As you mentioned, and it seems like, again, kind of in these animal models, where you look at a poorly adapted virus, so like an avian virus. So looking at flu, for example, this is work out of Tom Friedrich's group and Yoshikawa Welko's. If you look at poorly adapted virus, like an avian virus, flu virus, and you put into a mammalian host, and you kind of see whether there's transmission and so forth. You definitely have kind of a selective bottleneck, right? And so the ones that kind of establish in the recipients are the ones that are kind of better adapted, right, to replication in that host. For viruses that are, very well adapted, like seasonal influenza viruses in humans, it seems like most of those kind of mutations that you see would probably be deleterious, slightly deleterious, or you wouldn't even kind of, maybe even lethal deleterious. And so, the nonce genetic variation that you're actually seeing is going to be, just very slightly deleterious or maybe slightly advantageous, but it's going to be effectively kind of neutral. And if you look at, again, the work of Adam Loring's looking at seasonal influenza, you see kind of slight evidence for kind of, a little bit of purifying selection, but not kind of a huge amount. So the conclusion that basically there's a lot of genetic drift, going on within hosts and even kind of early on infection. I see where you're coming from in terms of, just the amount of virus that might be exhaled, and that can, so why are only kind of these, small number of viral particles seeding an infection when it could be more? I mean, for flu, it could be related to what are known as incomplete genomes. So what we know for flu is, it's a segmented virus that has 8 different gene segments, and a work out of from Chris Brooks's group, Jonathan Udall, and also Anise Lowen's group at Emory has really kind of established that most singly infected cells with kind of a single viral particle actually don't produce any progeny, because effectively there's a good chance for any one of those gene segments to effectively go missing, right? And then you can't actually produce, the cell can't actually produce viral progeny. So for seasonal flu, it makes sense to me that if there's a lot of kind of virus being kind of breathed out by a donor, and even if a lot of virus gets kind of deposited in the respiratory tract or the nasal tract of a recipient, that if those are kind of dispersed across cells, right, that just very few will actually kind of be able to go through that, even at that first replication cycle. And that would be kind of my expectation for SARS-CoV-2 and other even non-segmented viruses. It's obviously kind of not incomplete viral genomes there, but that it would just have kind of a very small probability of, even if kind of a viral particle lands on a cell, for it to actually find a cell that it can kind of infect. And actually, after that first, replication cycle or two, it seems like, the viral population can expand, but that's very kind of stochastic in the very beginning.
Well, does that mean that there's a whole lot of zeros that you don't see then, a very large amount of zeros?
Yeah, that would be my guess. Yeah, which is consistent with a relatively low R naught, right?
Yeah, but in the end, I don't think that would make that big of a difference, because the probability of a variant making it through is going to be dependent upon the prior probability of that variant in the population.
Sorry, can you elaborate on that again?
So you remember, Wright's basic theory on genetic drift, yep, which means that the probability of fixation of a variant is dependent upon its prior probability in the population. And so in this case, whatever the probability of the variants are and the viruses originally get transmitted, the chances of the ones that are going to be, were prior well off in the population is going to be determined by what they were in the previous population. So I don't think in the end, it won't really change the frequency of the variants of the ones that are doing a good job of infecting people. That's what I'm saying.
I mean, there's definitely, for SARS-CoV-2, there's definitely quite a bit of selection going on, right? As we know, so it's not just neutral genetic variation, right? As we've seen with kind of these variants of concern that have emerged and replaced almost I mean, now, in the US, what's the estimate right now? Is it greater than 90% of the viral infections are Delta? Something on that order, right? And so there's definitely kind of, selection going on at the population level. I think this kind of leads a little bit into kind of like, okay, well, where did those variants of concern kind of arise, right? And did those mutations arise kind of and spread within an individual first, right? Or did they arise kind of and fix through drift within an individual, and then that individual just had a higher, individual reproductive rate, right, than other individuals because they were able to infect more individuals because the virus population that they're infected with, has a higher kind of, replication rate. And so I mean, it seems like for some of these variants of concern, it gets into kind of chronic infections, right? So whether chronic infections are, these individuals who are immunosuppressed or immunocompromised who might get infected, and there might be enough time within those individuals for selection to occur, both for kind of a virus that has a higher replication rate, and then also might undergo antigenic evolution within that individual, and then might, some of these variants that have been that have kind of evolved within those long-term infected individuals might kind of pop back into the population level.
Yeah, I hope I didn't infer that I didn't think that was happening, because I do.
Okay.
Great, thank you. All right, so I think we have time for probably one last question. We have a question from Michael Reiskin. Michael, you can go ahead with your question.
Sorry, I get to speak at the beginning and the end of this. So I was actually curious about the connection between the tight bottlenecks and mutations, whether that was just probabilistic, so you're more likely to see one rise to fixation, or if there's a mechanistic way. Does the low diversity allow space for mutations to take hold in a new host? And maybe Michael has an idea about this.
Sorry, can you repeat that? I was answering someone's question in the chat.
And you can just kind of maybe give a little more details about that connection between tight bottlenecks and mutations. I wasn't sure if you were just saying it was like a probabilistic thing, so you're more likely to see a mutant become, rise to fixation, or if there's actually like a mechanistic way, the low diversity because of a bottleneck allows for kind of niche space maybe within the body for mutants to become fixed.
Yeah, I've always thought about it mostly just probabilistically, without, as Katia said, without like really rapid recombination. There's the higher the bottleneck, the less likely, just by probability, that a low frequency or that a novel variant will rise to fixation. By space, do you mean like physical space, like in terms of the cells in the respiratory?
We'll say competitive space. I mean, presumably they're competing against each other. So if for whatever, for entry into cells, for whatever makes one strain better than the other.
Yeah, I don't know. That's an interesting question though. Sorry.
That's okay. Maybe we'll
Anything
know in 10 years.
Anything you want to add, Katia? That's great.
No, I mean, I'm in agreement with Mike. I think it depends, if we have kind of these images of kind of fitness landscapes, right? So I think it also depends on kind of the virus that's kind of doing the infecting. Is it kind of towards a valley, in which case a lot of mutations might have higher fitness, right? And so might be kind of a higher chance that mutation that develops kind of initially might be advantageous, and so might be more able to kind of might even have an extra chance of kind of fixing, right? Versus something that a viral population that's already pretty well adapted to humans, right? So I think kind of patterns of which nucleotide variants actually fix, depends on how adapted kind of the virus is to the host population, or the host that it's infecting. And that might actually change over the course of, as we've seen for SARS-CoV-2, there's been adaptation to be able to, replicate within humans more effectively. So I think it's a kind of a changing pattern for SARS-CoV-2.
Okay, we're at the top of the hour, so we need to wrap up. Mike and Katia, that was fantastic. Really great to hear about your work, and this was, really interesting. And I like how you made it very interactive, and had some good discussion questions for us. I know there are a lot more questions people would like to ask, but we do have to wrap up. Before we do, I want to share our upcoming Club EvMeds. This is our webpage, of course, and the great talk we just heard. We have in a couple of weeks Molly Fox. We had to reschedule her, unfortunately, and she's going to be talking about Alzheimer's disease, a case of evolutionary mismatch. And then we have Sid Tacker from NC State. Well, that's actually coming up in November. We have a bunch of people in between there. The website's just not quite up to date yet. So stay tuned. We've got a lot of great speakers for you, a couple every month. We're going to aim for about one every 2 weeks. And we look forward to seeing you at Future Club EvMeds. Thanks again, Katia and
And
Mike.
thank you guys. It's great to see so many familiar faces.